All right, I want to run us through the body's nonspecific and specific immune responses. And we're going to pay special attention to the role of the antigen presenting cell in triggering the specific immune response. All right, so nonspecific and specific immune responses. The big difference here is that the nonspecific immune response, sometimes this is referred to as innate immunity. Um, this is built-in stuff, and it doesn't really matter what the invader is, the response is the same. That's kind of the key. So we have our first line, our first and second line of defense falls under the nonspecific mechanisms. So your first line of defense, of course, is your skin, which provides a barrier and separates what's outside of you from you, and then we have our mucous membranes, which secrete a lot of mucus to sort of trap potential invaders, and also some antimicrobial secretions as well. Then in our second line of defense, that's where our phagocytes, like our neutrophils and our monocytes, come into play, right? They gobble stuff up. They don't really care what it is necessarily. And it's not like they're going after a specific targeted invader. We have our antimicrobial proteins, like the complement cascade, which we'll talk about later, which falls under the heading of nonspecific. And then we have the inflammatory response, which we've talked a lot about. And the inflammatory response is the same. Doesn't matter what the injury or the invader is. It's the same response. So those are your nonspecific defense mechanisms. But then when we look at specific immunity, that's where this is a more targeted event. That's where the lymphocytes, both the B cells and the T cells, get kind of activated to some specific invader, be it bacterial or viral, and then the B cells are going to specifically produce antibodies to that antigen, and the T cells, once they're activated, they're going to go and they're going to bind to that infected cell, and they're going to try to lyse it or spit out some inflammatory or some degranulating or enzymes or something to kind of kill it, pop holes in it for the most part. So those are our specific. That's the specific response, right? So it's antigen specific. So for specific immunity, another way to think about this is it requires the recognition of self. So self, non-self. So there has to be some way for our lymphocytes to know that this is something that needs to go, right? And so sometimes the specific immunity is also referred to as adaptive immunity, because it's, we've evolved to become better at this. And, when we're faced with having to defend ourselves against all sorts of foreign invaders, this really works nicely, right? This is a strategy where we can develop specific antigens against all possible, specific antibodies against all possible antigens. And then we have this T cell situation where it's not mediated by antibodies, but it's still antigen specific. All right, so we'll kind of talk about this a little bit. So part of this recognition bit requires some ability to know this is something that should be here versus something that shouldn't be here, and that it relies on the presence of the presenting cell having basically this complex on them called the major histocompatibility complex, or also sometimes referred to as the HLA, human leukocyte antigen. So the MHC, or major histocompatibility complex, is common to the cells of the host. So the self, your cells have your MHC on it. The MHC is not the major histocompatibility complex is not an antibody, but it contains a bunch of, similar amino acid sequences that are going to resemble antibodies. And they're going to basically make the cells of a particular host unique to that particular person. So there's different types of these MHC classes or complexes on our cells. So on all of your own cells, you've got these MHC class 1, right? They are amino acid sequences. There's a variation, and they're going to be unique to you, unless, of course, you're an identical twin. Identical twins have this identical and major histocompatibility complex, which is interesting. All right, so again, this MHC then allows for specificity. It allows for the immune system's very accurate recognition of foreign invaders, because they're not going to have the MHC class 1 complex of the host. So how this all kind of gets started off is with a T helper cell. So a macrophage is going to gobble up, because it's a phagocyte, it's going to gobble up whatever, and it's gonna like partially digest that antigen that it's gobbled up, and it's going to kind of cleave the protein antigen, you know, and break it into peptides, and it's going to attach basically some of that antigen onto the presenting cells, the presenting cells MHC complex. All right, and then that presenting cell is going to then become the antigen presenting cell, right? The macrophage, you know, gobbles this up, presents onto the MHC complex, and that's now going to be called the antigen presenting cell, or sometimes it's called the dendritic cell. So these dendritic cells, or these antigen-presenting cells, sample little bits of antigen and place it in the MHC complex for a presentation. All right, so that presenting cell, that antigen-presenting cell, is then going to release some cytokines, interleukins, which we'll see interleukin 1, which will then produce interleukin 2, and other cytokines, which will then stimulate the further T cell proliferation. And B cell proliferation also. So I'm going to show you a picture of that in a minute. So this ability, because of the uniqueness of the MHC class 1s on all of our body cells, make it very easy to recognize all sorts of invaders. And then we're also going to see with specific immunity then the formation of these memory cells later on, which we'll remember them for later. All right, and I mentioned earlier that both B and T cells have memory. Okay, so kind of here's the story. We've got humoral immunity, and that relies on the antibodies being produced to protect the body. So what you see here on this picture, on the right side of the picture, you see the lymphoblasts from the bone marrow, or so basically bone marrow stem cells, which are going to either leave the bone marrow, go to the thymus, and mature and ultimately become T cells, or we're going to go the other direction. We're going to stay in the bone marrow. They're going to mature as B cells, and then once they are released, they're going to migrate to the lymph tissue, and they're going to hang out there. So they're going to hang out there until they get kind of told what to do, right? What comes next. And that's where we see in the middle, you see that macrophage gobbling up the antigen, processing that antigen, and then sticking the antigen on the MHC complex. So once it does that, we call it an antigen presenting cell, and now we're ready for basically that antigen presenting cell can stimulate the production of plasma cells to make antibodies or memory B cells, or also, can, cause the further differentiation and reactivation, for lack of a better term, of the T cells. So the what's happening on the right side of the slide is humoral immunity, where the antibodies are produced to protect the body. And on the very bottom of that, that's an antibody, that big Y-shaped structure. We'll talk more about that later. On the left side of the page, we've got the cell-mediated immunity, where lymphocytes are programmed to attack non-self cells. So this is cellular warfare, right? There's different types of T cells. There's the helper T cell. There's the memory T cell, which remembers it. There's the suppressor T cell, and then there's the cytotoxic T cell. Helper T cells are really important in the whole process, because when an antigen presentation activates a helper T cell, so once the macrophage eats the antigen, it becomes the antigen presenting cell, and that presentation of the antigen activates the helper T cell. The helper T cell is going to secrete cytokines, which are just, chemical messengers that stimulate B cell proliferation and ultimately antibody production, and also activate the T cells. So our helper T cells are really pretty specialized and sophisticated cells, and they really facilitate this whole immune process. Okay, what else do I want to see talk to you about that? Right, here's a little picture sequence of looking at a macrophage ingesting and then partially digesting some kind of microbial antigen. This particular one is a virus with T-dependent antigens, meaning that it's going to really facilitate the production of the T cell line. So then that macrophage gobbles up little bits of that antigen, presents it on the MHC class 2 proteins, which are the kinds that we see on the antigen-presenting cells. So that's going to then cause the helper T cell, call the helper T cell. The helper T cell is going to bind, and the helper T cell then is going to release cytokines that's going to stimulate the B cells and potentially the T cells. All right, so we're going to dive a little deeper here in the same topic and distinguish between cell-mediated and antibody-mediated or humoral immunity. We're going to look at the white blood cells, again, that are playing a role in each of these. We're going to talk about the role of the helper T cell. We're going to talk about the cytotoxic T cells, or the CD4 cells, and B cells and all that kind of jazz. All right, so cellular immunity, again, just the shakedown in terms of comparisons and compare and contrast. Cellular immunity uses cytotoxic or killer T cells. Those killer T cells, once they know what they're supposed to do, they'll directly attack foreign or damaged cells. Humoral immunity is going to use the B cell line. Most specifically, the B cells are going to differentiate into plasma cells to make antibodies. Once antibodies are made, it makes it easier to get rid of antigens from the circulation, which we'll talk about later, and ultimately they can be removed easier by the immune system. So helper T cells are, the role of the helper T cell in a general sense is to make both of these processes way more effective. All right, so here you have an antigen-presenting cell, right? The macrophage gobbled up the antigen, it presented the antigen, so it becomes an antigen-presenting cell. You see that the antigen is displayed on the class 2, MHC class 2 complex. You see that antigen-presenting cell then releasing interleukin, which is a cytokine, interleukin 1, and then the helper T cell is going to release interleukin 2, which is going to stimulate the proliferation of both the cytotoxic T cells and the B cell production, differentiation, or B cell differentiation into plasma cells and ultimately memory cells. So that's the role of the helper T cell. It facilitates both of those arms of immunity, both cell-mediated and humoral immunity. It makes the whole system way more robust. It makes it much more efficient, and makes the immune system, as a consequence of that, way better equipped, much better equipped to deal with these invaders. All right, so again, the sequence in cell-mediated immunity, this is the one that uses the T cells. The macrophage is going to engulf the foreign invader. Parts of the invader, the antigen, are going to combine with the MHC complex. They're going to move to the surface. Now we're going to call that cell an antigen-presenting cell. The antigen-presenting cell is going to stimulate the helper. So the antigen-presenting cell is going to release interleukin 1, which is going to call the helper T cell. The helper T cell will release interleukin 2, which will make the cytotoxic T cells divide to make more cytotoxic T cells and more memory T cells. So here's another picture. This is a picture of an infected cell, and then that thing that's stuck to it, that gray color, that's a cytotoxic T cell. And in the granules there's different types of chemicals that will destroy that infected cell. In this case, that cytotoxic T cell is going to spit out something called perforin, which perforates the infected cell's membrane, pops holes in it, which is going to cause that cell to swell, right? When you pop a hole, you make the cell membrane, you disrupt the cell membrane. That's going to allow for water to rush in there, and that thing's going to blow up, which you can see happening there. So that's cellular warfare. It's awesome and really effective, right? We really need that to happen. All right. So cells, these helper T, or pardon me, these cytotoxic T cells recognize all sorts of things as damaged or foreign, including things that maybe it shouldn't. So grafts, you know, when you take a bit of tissue from one place and stick it someplace else, that sometimes the immune system will recognize that as foreign and go after it. Cancer cells ideally will get recognized as damaged or foreign, but unfortunately sometimes they go under the radar because they are our own cells, which mean they oftentimes have our MHC class 1 complex on them, so our immune system doesn't always notice them, but they do act kind of weird, and so that helps. If you have a cell that has been infected with TB or any kind of fungus or any viral infections, contact dermatitis is an example of cell-mediated immunity. All right, humoral immunity. Now this is blood-borne immunity, or humoral, what was the other use I word? I used another word to describe it earlier. So this is where we start to see antibodies. So I think we all know about antibodies and antigens, but just in case we don't know these terms, I should probably define them for you. So an antigen, so the two terms are antigens and antibody. An antigen is any substance that can combine with a receptor on a lymphocyte, keeping in mind that each lymphocyte's antigen receptor is unique, and the interaction with the foreign antigens will stimulate an immune response. So it's a foreign invader that's going to stimulate an immune response in the host. An antibody is a defense mechanism, a defense molecule. An antibody is produced by the body in response to the antigen. So there's different types of antibodies, which we're going to talk about in a little while. So these are, this falls under the heading of specific immunity, right? Which is, or sometimes it's called adaptive immunity, which is the body's adaptation to these antigens. All right, so antigens are protein molecules. They're pretty big. Some are bigger than others. We'll go through the different types of antibodies at another time. All right, so here's your homework question. I would like you to describe for me the normal sequence in the activation of humoral immunity, differentiating the primary and secondary response in terms of time of onset and maximum antibody production. So this is your homework question for Unit 5. So here we have it. This is the sequence of events. This is humoral immunity, right? Humoral immunity is using the B cell line. So it's going to start off the same way. The macrophage, or a B cell, is going to engulf a foreign invader. The macrophage, or the B cell, is going to gobble up that foreign invader and bits of the protein sequence, and they're going to present bits of that protein sequence on the MHC class 2 complex on the surface of the cell. We're going to again call this thing, if it's a macrophage, we're going to call it an antigen presenting cell. That antigen presenting cell is going to release some interleukin 1, which will stimulate the helper T cell to bind. The helper T cell will release interleukin 2, which is going to make the B cells divide into plasma cells, or sometimes they're called effector B cells. That's the same. The plasma cells make the antibody. And then we've got the memory cells. The memory cells are going to remember that antibody for later. So the memory B cells play a very important role because they are going to, again, they're going to enable a much quicker response to that same antigen in the future. Again, these things hang out in our lymphoid tissue germinal centers, and then they circulate in the blood, to various places. Okay. What else do we want to see here? Okay, so here's a picture. We've got a macrophage binding to a helper T cell, same thing we saw before, but this time the helper T cell is going to interact with a B cell. That B cell is going to display the same foreign antigen as the class 2 MHC proteins, and that B cell then is going to develop into a plasma cell, which is the antibody secreting cell, right? So it's going to make antibodies, which you see there, those Y-shaped things. And then we're also going to make memory cells. So here's a plasma cell. It's a pretty big cell. That's the one that makes the antibodies. All right, so Next, we want to distinguish between the human's body's primary and secondary immune response. This is pretty straightforward. So the primary and secondary immune responses are the way the body responds to antigens on the first exposure, and then any any subsequent exposure would be secondary. The first exposure is primary. So in that picture you see down there at the bottom, you see antigen A being the body being exposed to antigen A, and you can see the time in weeks on the bottom, and then the antibody titer in the blood, the serum antibody titer. So the serum antibody titer in this case, because it's a primary exposure, you see that the serum antibody titer doesn't even get going, doesn't even really start raising until 2 to 4 weeks is when it really peaks, and it's really not that good of a peak. But if you see just after 7 weeks, you see another exposure to antigen A, same antigen. Check out that secondary response, right? It's like way faster and way more robust. That's a secondary response. So again, the primary response is the first exposure to the antigen. It usually takes a couple weeks before the antibody titer reaches efficacy, where it tops out. So it peaks and then it goes down again. The secondary response would be the repeat exposure to the same antigen. It has to be the same antigen. If it's a different antigen, it's not going to evoke the same kind of a response. But if it's the same antigen, you've got memory. They already know this. They've seen this. They've played this game before. That antigen has already been marked as something that you definitely don't want there, and those memory cells are going to respond immediately to this, and they're going to produce a whole bunch of antibodies, right away, and that's why you see that huge peak there. So the secondary response usually is quicker, like 1 to 3 days, and produces a much greater amount of antibody. Mostly it's going to be the IgG type of antibody, which we'll talk about later, but sometimes IgA and IgE. And again, the major reason for this robust response is the proliferation of the B cells, right? They proliferate during the primary response, and then they hang around and they wait, and they're ready. And when that particular offending antigen shows up again, they are gonna go crazy, right? And they're gonna produce a whole bunch of antibodies in response to that. So that's that. All right, here's a picture of an antibody. So an antibody is shaped kind of like a Y, the letter Y, and it has a couple binding sites, which are important to note. It has a binding site for the antigen, pardon me, and then it has a binding site. We've got a couple binding sites, which we'll run through, which are not important to remember, but I just want to tell you all about them. All right, so we've got the basic structure of the immunoglobulin and the 5 different kinds of immunoglobulins. That's where we're at here. So the structure of a antibody molecule, again, it's shaped kind of like the letter Y. There are a whole bunch of specificities of the antibodies that determine kind of the different portions of the antibody molecule. There are basically 2 sections, the 2 arms of the Y. Those are called the FAB, F-A-B sections, and they contain what we call a heavy and light chain. The variable portion is at the free end of the FAB arm. For whatever reason, I don't have a cursor on here. So the purple part is the FAB part. At the bottom, the pink part that's kind of hanging free, that's the variable portion. You don't need to remember of that, but that's kind of the story. So what has to happen here is there's a antigen binding site, which is up at the top, kind of where those S's are. There's also a complement binding site, and then there's a receptor binding site. The and again, you have these heavy and the light chains, which is not important. Let's see if I have a better picture. Oh, I guess I don't have another picture. All right, so the types of, there's 5 types of antibodies, major antibody groups. They can be abbreviated by the mnemonic GAMED, G-A-M-E-D, if you like mnemonics. That would be IgG, IgA, IgM, IgE, and IgD. Again, the isotype, the particular type of immunoglobulin is going to be determined by the kind of that consistent portion, the FC portion of the immunoglobulin. All right, let's go through. I'm going to put them all up and I'll talk about them. Okay, IgG gets first billing because IgG is kind of the classic immunoglobulin. IgGs are the most common in the blood. So if you're measuring immunoglobulins in the blood, chances are you're measuring IgG. IgM is the main antibody in the primary immune response. It's big, that's why it's called, it's like the magnum. The IgM is the biggest one. It's also really good at activating complement, which we're going to talk about in a minute. IgA is the main antibody in secretions. So you see tears, saliva, mucous membrane, colostrum. IgA plays a very significant role in the first line of defense at the mucous membranes. And anything that's got mucosa, IgE. So you see it a lot in the gut. We measure it in people with GI issues. Sometimes if they have not enough IgA, they're more prone to infections. IgE is primarily found in allergy and worm infestations, very similar to the eosinophils. If you remember, we said that the IgE immunoglobulin binds to eosinophils, also basophils and mast cells. And it's a big player when somebody has allergic hypersensitivity reactions, which we'll talk more about later. IgD is the only antigen receptor on B lymphocytes. We don't know a lot about IgD, other than it activates B cells. What else do we need to say? I think that's probably good. So what you want to remember, I would like you to remember which one's the biggest one, which one's the most common in blood, the allergic piece of IgE, and which one's the most common in secretions. All right, so I said the IgD, we're not totally sure what's going on with IgD, but it plays a role in B cell differentiation. Here's a little chart that gives us some kind of good information in terms of size. So you've got IgG, IgA, IgM, IgD, IgE, molecular weight. You can see by far the IgM, is the biggest by a long margin. Most of them are pretty much all kind of in the same range. The serum concentration you can see for IgG is 1,200 milligrams per deciliter. That's by far and away the most. The half-life, IgG hangs around for about 23 days. So IgG hangs around. The ability to fix complement, which we'll talk about in a little while. The one that's best at that is IgM, which we said earlier. IgG is okay. Those are the only two that can fix complement. And IgG is the only one that can cross the placenta. All right, so when we talk about antigens and antibodies and how they interact with one another to get stuff out of the allow the immune system to sort of take care of it. That's kind of what we want to look at here. So these immune complexes, once the antigen binds, the antigen antibody binds together and they form a complex. Those complexes are pretty big, right? Because the immunoglobulin is pretty big, and it's got all this antigen stuck to it in various forms. Oftentimes, those complexes can precipitate. and that allows the immune system to kind of find them. So things that normally would be soluble can precipitate when they're stuck to an antibody, depending on the ratio of antibody to antigen. And that allows for the, again, for the immune system to sort of to find it. Oftentimes when an antibody binds an antigen, it neutralizes the antigen, so it sort of takes it neutralizes the toxin. Agglutination means when they stick together. So when something agglutinates, it gives, again, it makes it a lot easier for the immune system to find it because you've got this whole antigen antibody kind of clump. I mentioned the precipitation. The precipitation is another way that, again, something that would normally be soluble in water is able to kind of precipitate out and allows the phagocytic cells to find it easier and, eat it up, get rid of it. That's the goal here, is to clear these things. Opsonization. Opsonization is an example of when they kind of all stick together. I'm going to show you a picture in a minute. It will make more sense. And then complement fixation. I'm going to talk about complement in just one second. So here's a little picture of neutralization, the antibody binding onto the virus or the bacteria, neutralizing that toxin. Then you see a kind of a whole cluster of antibodies sort of stuck together by the bottom of the Y, and then the bacteria stuck to the binding sites. So that's agglutination of the particles. Again, the immune system's going to be able to find that easily. Then we take something that would normally be soluble and make it insoluble, so the immune system can find it. And agglutination is a kind of a bigger clump. I thought I had a picture of agglutination, but I guess I don't. When something agglutinates, it basically forms this clump. When an antigen-antibody reaction occurs, these things clump together, and again, that makes it easier for the immune system to clear it. Here we see IgG tends to aggregate, meaning it tends to stick together. All right. I'm going to just talk about complement, and then I'll finish this video and start another one later. So complement is interesting. Complement falls under our nonspecific immune response. If you remember from the very beginning of our first lecture video for this unit, we talked a little bit about the nonspecific responses, and I mentioned that complement falls into that category. So this complement system, I should say, is pretty interesting. And what happens with complement is that we've got these proteins that are activated in sequence. That's kind of the important thing to know about complement. So with complement, we've got this series of proteins that get activated in a sequence, and ultimately it's going to lead to is it's going to lead to the lysing of cells, invaders. So there's at least 15 proteins. Most of these proteins are enzyme precursors, which are produced mostly in the liver. The proteins are then found in the blood, in the serum, and when they're activated, they undergo this chain reaction, these chain of conversions that are really important in the immune response. So we have the classical pathway, which you can see on the top. In the classical pathway, the factor that activates the complement cascade is an antigen-antibody complex. So if you see up there at the top, you see the antigen-antibody complex. In the alternate pathway, which is down below that dotted line, in the alternate pathway, complement activation does not require the interaction of complement with the antigen-antibody complex, but instead it requires oftentimes it's bacterial lipopolysaccharide or some kind of endotoxin or other bacterial substances. So a toxin potentially produced by the bacteria or other bacterial substances that can activate the alternate cascade. Sometimes IgA, I should say, can alternate or IgA aggregates can alternate the alternate pathway. So kind of the key to the alternate pathway is that C3 component of complement getting split into the C3b and the C3a components, which you can see there. And those components kind of have different functions. So the C3b cleaves into what we call C5 and then into C5a and C5b. You don't need to remember that. But basically what that does is so one of those, when the C3b cleaves, one effect is to cause the cell to basically pop holes in the cell membrane, and the infected cell membrane, and the cell swells and bursts. The other side of this forms a membrane attack complex that, goes and starts attacking the membrane. The net effect of both of these is to destroy these cells. And again, it doesn't really matter what type of bacteria, what type of antigen-antibody complex that forms to the C1 complex, or what type of endotoxin potentially starts this whole thing off. So that's what makes it, non-specific. So what you're going to want to remember is, so the complement cascade is activated during immune reactions. Usually it's IgG or IgM, sometimes IgA aggregates for the alternate pathway. The proteins, the proteins are inactive in the blood, but they get activated as a result of one of these two pathways. So the proteins start from C1 to C9. You don't need to remember all of those. Don't memorize that picture. But just kind of know that the net effect of this complement cascade is to damage the cell and also any other, and kind of like quells the inflammatory, or I should say, propels the inflammatory response once it's activated. So that's complement. So what I mean by that is some forms of complement can actually stimulate an acute inflammatory response, including the release of histamine and mast cells from basophils. So this will make our blood cells more leaky, it will cause more inflammatory cells, as well as that complement protein entering into damaged tissues. So it's a lot. It's a general inflammatory response that's pretty complex. And it kind of just both destroys cells and, kind of amplifies this inflammatory kind of response as well. So that's complement. All right, so I'm going to show you that picture. So here we have the activation of the complement cascade, and then ultimately the complement proteins are going to be activated. They're going to pop a hole in the cell, form a membrane attack complex, and also cause that cell to lyse. All right, there's some other chemical mediators that we don't really talk about them, but I wanted to introduce you to these because they'll come up. Like we need to know more about this, right? And the more we learn kind of like on the molecular level about inflammation, the more complicated it gets. So these are chemical mediators. They perform a variety of functioning, some of which is cell signaling, communicating between cells, and some causing cellular damage. So I just made a little chart here. You don't need to memorize these, but I want you just to know about them. So the kinins, and these are things you'll read about in literature. The kinins, when they're released, they cause vasodilation. They cause an increase in capillary permeability, and they can cause pain. The prostaglandins are a group of lipids. So they're basically fatty acids that can be released from damaged cells, including mast cells. They have a whole bunch of different jobs. One of which is to enhance the inflammatory response by, again, increasing vascular permeability. They also seem to have some chemotactic properties, which means they call neutrophils towards the wound site, and they can also stimulate free nerve endings, free pain endings. Leukotrienes are a group of lipids also derived from mast cells and basophils. that cause contraction of bronchial smooth muscle, and they have a role in the development of inflammation. Some of the drugs we used for like allergies, when somebody's airway constricts, they utilize those leukotrienes, block those leukotrienes, I should say. Cytokine, that's a very general term, usually signifying some kind of protein hormone that affects cells that are near the site of the origin of the cytokine. So there's all sorts of them, lymphokines, monokines, interferons, interleukins. Those are all cytokines. They're produced by macrophages and also activated T lymphocytes. They stimulate the activation of B cells and T cells and also facilitate the communication, like I said, between cells. They're short distance communicators. They're very definitely involved with fever, inflammation, and calling a chemotaxis, calling of white blood cells to the area, which is what leukocytosis means. Tumor necrosis factor, really, it's a cytokine that's very active in the inflammatory and immune response. It's really good at stimulating acute inflammation, and you can see that it stimulates fever, chemotaxis, mediator of tissue wasting, stimulates T cells, mediator in septic shock. And then chemotactic factors are going to be the ones, like I said, that call white blood cells to the area. All right, so I'm going to stop there. We'll pick up this with Objective 10 on another video.